Family trees might seem like dusty old charts, but what if they're actually the key to tracking deadly diseases, solving crimes, and even saving endangered species? Today, we're exploring how these evolutionary maps are revolutionizing science in ways nobody expected.
That's such an interesting point about disease tracking. You know, during the COVID pandemic, scientists used these evolutionary trees to predict which variants might become dangerous next.
Well, here's what's really fascinating. These phylogenetic trees helped researchers track mutations in real time, almost like having a GPS for virus evolution. They could actually see how the virus was changing and spreading across continents.
Hmm. And it's not just for viruses. I heard they're using similar techniques in forensic science now.
Oh, you're going to love this. Forensic scientists can actually use DNA analysis to create evolutionary trees that link evidence from crime scenes. It's like CSI meets Charles Darwin.
So, what happens when they find species that look identical but turn out to be completely unrelated?
That's where it gets really wild. Take dolphins and sharks. They both have that sleek, streamlined body, perfect for swimming, right? But here's the mind-blowing part. They're about as closely related as we are to fish.
Well, that's what scientists call convergent evolution, when different species independently develop similar features because they face similar environmental challenges.
Exactly. And you know what's even more fascinating? Scientists have found that this happens way more often than we originally thought. Like how both bats and dolphins independently developed echolocation.
That makes me wonder, how do researchers tell the difference between features that come from common ancestry versus those that evolved separately?
So they use this really clever approach called the principle of parsimony, basically looking for the simplest explanation that fits all the evidence. But here's where it gets interesting. Sometimes the simple answer isn't the right one.
That's when they bring in those more sophisticated methods like maximum likelihood analysis, right?
You got it. And these tools are becoming incredibly important for real-world applications. Like, did you know conservation biologists use phylogenies to decide which endangered species to prioritize for protection?
Well, that makes sense. You'd want to save species that represent unique evolutionary lineages.
Exactly. Think of it like preserving rare books. If you had to choose between saving the last copy of a unique manuscript or one of many copies of a common book, you'd probably choose the unique one.
And I've heard they're using similar approaches in agriculture to develop better crop varieties.
Oh man, this is where it gets really practical. By understanding the evolutionary relationships between different varieties of crops, scientists can predict which crosses might produce drought-resistant plants or higher yields.
That could be crucial for dealing with climate change, couldn't it?
Absolutely crucial. And here's a wild statistic. Scientists estimate that understanding these evolutionary relationships has helped increase crop yields by up to 30% in some cases.
That's incredible. It's like having a roadmap for future food security.
And speaking of roadmaps, these tools are also helping us understand how diseases might evolve in the future. Like, researchers can actually predict which flu strains are most likely to cause the next outbreak.
Well, that must be incredibly valuable for developing vaccines.
You know what's really mind-blowing? These evolutionary trees have helped reduce vaccine development time by up to 50% in some cases. It's like having a crystal ball for disease evolution.
So these tools are literally saving lives while also helping us solve crimes and feed the world?
That's exactly right. And we're just scratching the surface. Scientists are now using phylogenies to understand everything from ocean acidification impacts to the spread of antibiotic resistance.
It's amazing how something that started as a way to understand evolution has become such a powerful tool for solving modern problems.
And here's what really gets me excited about the future. As our genetic sequencing technology gets better and better, these tools are becoming even more powerful. We're talking about being able to track changes in ecosystems in real time.
That could be game-changing for environmental protection.
Well, it already is. Scientists recently used phylogenetic analysis to identify previously unknown relationships between species in the Amazon rainforest, helping to protect entire ecosystems rather than just individual species.
It sounds like we're entering a new era of scientific discovery.
You know what? We really are. These evolutionary trees aren't just about understanding our past. They're becoming essential tools for solving some of the biggest challenges we face today. From fighting diseases to protecting endangered species to feeding a growing population, understanding these connections is more important than ever. Ever wonder how scientists can trace the entire history of life on Earth? It turns out they're using something that looks a lot like your family tree, but on a mind-blowing scale that connects every living thing that's ever existed.
That's fascinating. And these evolutionary trees, or phylogenies, are actually powerful tools that help us understand everything from disease outbreaks to conservation.
Let me break this down in an interesting way. When scientists create these trees, they're basically playing detective with DNA, physical characteristics, and fossil evidence to map out how species are related to each other.
You know what I find really compelling? How these trees can show us that appearances can be totally deceiving when it comes to evolution.
Oh, exactly. Take dolphins and sharks, for example. They look similar and both live in the ocean, but here's the wild part. Dolphins are actually more closely related to cows than they are to sharks.
That's such a perfect example of how these evolutionary relationships can surprise us. Could you explain how scientists figure out these connections?
Well, it all comes down to something called branch points, or nodes in scientific terms. Think of it like this. Every time you see a split in the tree, that represents a moment when one species divided into two different evolutionary paths.
So it's kind of like tracing your ancestry back through time, but for entire species?
Exactly right. And here's where it gets really interesting. Scientists use something called clades to group organisms together. A clade includes an ancestor and all its descendants, like saying everyone in the mammal family or all species of pine trees,
That makes me think about how some features might look similar but have completely different origins.
right? This is where we get into homology versus analogy. Take wings, for example. Birds and bats have homologous wings because they evolved from the same ancestral arm structure. But bird wings and butterfly wings, those are analogous. They serve the same purpose but evolved independently.
Hmm. So it's like nature came up with similar solutions to similar problems, but through different paths?
Precisely. And what's really cool is how scientists use something called the principle of parsimony to figure out which evolutionary story makes the most sense. They look for the explanation that requires the fewest evolutionary changes, kind of like choosing the simplest route on a map.
That's such a clear way to think about it. But how does this knowledge help us in the real world?
Oh, this is where it gets really exciting. These evolutionary trees are crucial for tracking disease outbreaks. During the COVID-19 pandemic, scientists used phylogenetic analysis to track different variants as they evolved and spread globally.
That's incredible. So it's not just about understanding the past?
Not at all. These trees help us identify which species need urgent conservation protection, improve crop breeding programs, and even understand how antibiotic resistance develops in bacteria. It's like having a roadmap that shows us both where we've been and where we might be heading.
Well, that really puts things in perspective. We're using evolutionary history to solve modern problems.
And here's something mind-blowing. Every new species we discover and every genome we sequence adds another piece to this enormous puzzle. It's constantly evolving, just like life itself.
So these phylogenetic trees are really more like living documents than static diagrams.
Exactly. And they're helping us prepare for future challenges too, like predicting how species might respond to climate change or how diseases might evolve. It's pretty amazing how understanding our past can help us shape our future.
That's such a powerful way to think about it. These aren't just academic exercises, they're practical tools for solving real-world problems.
And when you really think about it, we're all part of this massive interconnected tree of life. Every species alive today represents the success of billions of years of evolution. Pretty incredible, right? What if everything we think we know about species is completely wrong? Today we're exploring how the simple concept of different animals being different species is actually one of biology's biggest puzzles.
That's such an interesting point. Most people think they can easily tell species apart. but scientists have been debating this for over 200 years.
You know what's fascinating? There are at least 26 different ways that scientists try to define what makes a species. The most common one is about breeding, but that doesn't even work for about 90% of life on Earth.
Hmm, that's because most organisms don't reproduce sexually at all, right? Like the billions of bacteria species out there.
Exactly. And here's something mind-blowing. We estimate there are around 8.7 million species on Earth, but we've only identified about 1.2 million of them. That means we're trying to create rules for millions of species we haven't even discovered yet.
Well, that certainly complicates things. So, how do scientists actually approach this problem?
They use what they call the biological species concept as their main tool. It's all about whether organisms can successfully mate and produce fertile offspring. But here's where it gets interesting. This definition fails spectacularly in so many cases.
Like with those bacteria you mentioned earlier?
Right, and get this, about 80% of plant species can actually form hybrids with other species. According to the strict biological species definition, they should all be considered the same species, which is clearly not right.
That's wild. So, what other ways do scientists try to classify species?
Well, there's this fascinating thing called reproductive isolation, nature's way of keeping species separate. It happens in two main ways, prezygotic and postzygotic barriers. And some of these barriers are absolutely incredible.
You know, I've heard about those terms, but never really understood what they mean.
So, prezygotic barriers are like nature's bouncers. They prevent mating from happening in the first place. For example, did you know that some species of fireflies have completely different flash patterns? If they don't match the right pattern, they don't mate.
That's like having a secret password to get into an exclusive club.
Exactly. And then we've got postzygotic barriers, which are like relationship problems that show up after the fact. The most famous example is probably mules. They're healthy and strong, but 99.9% of them can't reproduce.
Speaking of reproduction, how do new species actually form? I mean, this whole process of speciation must be pretty complex.
Oh, it's fascinating. There are two main ways, allopatric and sympatric speciation. Allopatric is more common. It happens in about 80% of speciation events. Think of the Galapagos Islands, where one species of finch evolved into 13 different species.
That's amazing. So, physical separation really drives species formation.
Usually, yes. But then there's sympatric speciation, which is like watching evolution happen right in front of us. For instance, there's this apple maggot fly that used to only eat hawthorn fruits. But in the 1800s, some of them switched to eating apples. Now they're becoming two different species, all while living in the same area.
That really challenges what we think we know about how species form.
And here's something even more mind-bending. We're actually watching new species form right now. Climate change is forcing some populations to adapt so quickly that they're splitting into different species. Some scientists estimate this is happening at about 3 times the normal rate.
That's both fascinating and a bit concerning. It really shows how our actions are affecting evolution in real time.
Absolutely right. And you know what's really interesting? Some scientists now think we need to completely rethink how we define species. With new genetic tools, we're finding that many species regularly exchange genes with other species, and it's called hybridization.
So, the boundaries between species are actually more fluid than we thought?
Exactly. Recent studies suggest that about 10% of animal species and up to 25% of plant species regularly hybridize in nature. It's like nature doesn't really care about our neat categories and definitions.
That really puts things in perspective. We're trying to put rigid labels on something that's naturally very fluid.
Well, I think that's what makes this whole topic so fascinating. Every time we think we understand what makes a species, nature shows us something new that breaks all our rules. It's a humbling reminder of just how complex and amazing life on Earth really is. Evolution is often called survival of the fittest. But what if that common phrase is completely wrong? Today, we're exploring how random chance and unexpected forces shape life on Earth in ways most people never imagine.
That's such an interesting point. The mechanisms of evolution are far more complex than most people realize. It's not just about being the strongest or fastest.
Let me share something fascinating. There are actually 5 distinct mechanisms that drive evolutionary change, and natural selection is just one of them. Want to know what really blew my mind when I first learned about this?
Well, don't keep us in suspense. What's the unexpected part?
So get this. Sometimes totally random events can have a bigger impact on evolution than any survival advantage. It's called genetic drift, and it's like nature's version of rolling dice. Think about cheetahs. Their limited genetic diversity today isn't because of survival advantages, but because their population crashed at some point, leaving only a tiny group of survivors.
Hmm, that's really interesting. So you're saying chance events can sometimes override natural selection?
Exactly. And here's another mind-bending example. The founder effect. Imagine a small group of birds gets blown off course to an isolated island. Whatever random genetic traits those specific birds happen to have become the foundation for an entirely new population. It's like starting a recipe with just a few random ingredients from the original kitchen.
That makes me think about how these random processes must interact with the other mechanisms of evolution. What are the other forces at play?
Well, besides genetic drift and natural selection, we've got mutations, which are like nature's typing errors in DNA, gene flow, which happens when populations mix and share genes, and non-random mating, where organisms are picky about their partners. It's like having 5 different artists all working on the same canvas.
You know what's really counterintuitive about this? The whole survival of the fittest concept seems to miss the mark entirely.
Oh man, exactly. Fitness, in evolutionary terms, isn't about being the strongest or fastest. It's about how well an organism's traits help it survive and reproduce in its specific environment. Sometimes being smaller or slower might actually be the better strategy.
So, how does this play out in real populations? Are there different patterns we can observe?
There are 3 fascinating patterns of natural selection. First, there's directional selection, like when drought causes birds with larger beaks to survive better because they can crack tough seeds. Then, there's disruptive selection, where both extremes of a trait are favored. Imagine birds with either very small or very large beaks doing well, while medium-sized beaks are disadvantageous.
And isn't there a third pattern where being average is actually the best strategy?
Yes, it's called stabilizing selection, and human birth weight is the perfect example. Babies that are either too small or too large tend to have more health problems. So there's this natural pressure keeping birth weights in a middle range. It's like nature's version of the Goldilocks principle. Not too big, not too small, but just right.
That brings up an interesting question, though. If these mechanisms are constantly working, why aren't all species perfectly adapted by now?
Well, here's the thing. Evolution can only work with what it's got. It's like trying to renovate a house while living in it. You can't tear everything down and start from scratch. Plus, environments are constantly changing, so what's adaptive today might not be tomorrow, and then there's the issue of trade-offs.
Like the peacock's tail, right? Beautiful but potentially dangerous?
Exactly right. Those magnificent feathers are great for attracting mates, but they also make the birds more visible to predators and harder to fly. Evolution is constantly balancing these competing pressures. And here's what's really mind-blowing. These mechanisms are happening all around us all the time.
That really puts things in perspective. Evolution isn't just ancient history, it's an ongoing process.
And that's why understanding these mechanisms is so crucial. It helps us understand everything from antibiotic resistance to climate change adaptation. These aren't just academic concepts. They have real-world implications for medicine, conservation, and our understanding of life itself.
So, in the end, it's really about understanding how all these forces work together to shape life on Earth.
You know what's amazing? We're still discovering new aspects of how these mechanisms work. Every new study adds another layer to our understanding of this incredible process that's been shaping life for billions of years and continues to shape it right now in ways we're just beginning to understand. Ever wonder why some genetic traits seem to vanish from populations while others stick around for generations? The mathematics behind this evolutionary dance is more fascinating than you might think.
Well, that's quite a puzzle to unpack. The Hardy-Weinberg equilibrium actually gives us a mathematical framework to understand when evolution is or isn't happening.
You know what's really interesting about this? Scientists discovered that if certain conditions are met, like having an infinitely large population with completely random mating, gene frequencies should stay perfectly stable.
Hmm. But those conditions never actually exist in nature, right? That's what makes this principle so useful as a baseline measurement.
Exactly. It's like having a perfect control group. When we see populations that don't match these predictions, we know something evolutionary is happening.
So, how do we actually measure these genetic changes mathematically?
Well, it comes down to two simple letters, P and Q. If P represents the frequency of a dominant allele, and Q represents the recessive one, they always add up to 1. Then we can use P squared plus 2, PQ plus Q squared equals 1 to predict genotype frequencies.
Oh, let me share an example that makes this clearer. If we look at a population where 16% show a recessive trait, that means Q squared equals 0.16.
Right, and from there, we can work backwards. The square root of 0.16 gives us Q equals 0.4, which means P must be 0.6. Then, we can predict that 36% should be homozygous dominant, 48% heterozygous, and 16% homozygous recessive.
And here's where it gets really interesting. If we count the actual population and get different numbers, we know evolution is happening.
Exactly. Like if we found 60% showing the dominant trait instead of 36%, something must be disrupting the equilibrium. Maybe natural selection is favoring that trait, or maybe there's non-random mating happening.
You know, this reminds me of how antibiotic resistance spreads in bacteria. When we use antibiotics, we're creating intense selection pressure that completely violates Hardy-Weinberg equilibrium.
That's such a crucial real-world application. Understanding these principles helps doctors and researchers predict how quickly resistance might spread through bacterial populations.
And it's not just about medicine. These principles are vital for conservation biology, too.
Well, speaking of conservation, wildlife corridors are a perfect example. By connecting isolated populations, we're managing gene flow to prevent genetic drift. It's like applying Hardy-Weinberg principles to save endangered species.
That's fascinating. And it shows how theoretical principles can have practical conservation impacts.
You know what still amazes me? There are 5 main mechanisms that can disrupt Hardy-Weinberg equilibrium: mutations, gene flow, genetic drift, non-random mating, and natural selection. Each leaves its own unique signature in the population.
Like genetic fingerprints that tell us exactly how evolution is working in real time.
And these principles apply everywhere, from understanding how climate change might affect species adaptation to tracking the spread of beneficial traits in crop plants.
Well, that brings up an interesting point about human populations, too, though we have to be careful about how we apply these principles there.
Absolutely right. We need to remember that Hardy-Weinberg is a tool for understanding population genetics, not for making value judgments about traits.
So, what's the biggest takeaway for someone just learning about these principles?
I'd say it's that evolution isn't just some vague force of nature. It's something we can actually measure and predict mathematically. Hardy-Weinberg gives us a way to spot evolution in action by showing us when populations deviate from theoretical expectations.
And those deviations always tell us something interesting about what's happening in nature.
Looking ahead, these principles will become even more important as we face challenges like antibiotic resistance and climate change. Understanding how populations evolve helps us make better decisions about everything from medical treatments to conservation strategies.
That's quite a legacy for what started as a simple mathematical model.
Well, that's the beauty of science. Sometimes the simplest principles help us understand the most complex phenomena in nature. Thanks everyone for joining us on this journey through the mathematics of evolution. Evolution seems like it should be simple, survival of the fittest, right? But what most people don't realize is that individual organisms don't actually evolve at all. It's entire populations that change over time.
That's such a fascinating point. The DNA we're born with stays the same throughout our lives. What we're really talking about is how genetic variations spread through populations across generations.
I used to think that when someone got stronger from working out or learned a new skill, that was evolution. But now I understand that's not how it works at all.
Well, here's what makes it even more interesting. Those physical changes you're describing are just adaptations within your lifetime. True evolution requires genetic changes being passed down through generations.
Speaking of genetic changes, let's break down how variation actually happens in populations. There are three main ways, right?
Mm-hmm. First, we have mutations, which are random DNA changes, then genetic recombination during reproduction, and finally, gene flow when organisms move between populations.
I've heard people compare mutations to typos in DNA. Some make things worse, some might actually help, and many don't really matter either way. But what determines if a mutation is helpful or harmful?
That's exactly the key point. It all depends on the environment. Like, imagine a mutation that makes an Arctic hare's fur darker. In a snowy environment, that's terrible for survival. But if the climate becomes more rocky, suddenly that same mutation could be an advantage.
So this is where the math comes in, right? We can actually track these genetic changes using some pretty straightforward equations.
Yes, the basic formula is P plus Q equals 1, where P represents dominant allele frequency and Q is recessive. Want to see how this plays out with real numbers?
Definitely. Sometimes these concepts make more sense with actual examples.
Well, let's say we have a population where 70% of alleles are dominant. That means P equals 0.7 and Q equals 0.3. When we do the math, we get 49% with two dominant alleles, 42% with mixed pairs, and 9% with two recessive alleles.
You know what's wild? How we can actually see evolution happening in real time with things like antibiotic resistance. It's like watching these mathematical principles play out in the real world.
That's a perfect example of human-induced evolution. When we use antibiotics, we're creating conditions where bacteria with resistant genes are more likely to survive.
And this applies to so many other situations too, like pesticide resistance in insects or how urban wildlife adapts to city life. It's kind of sobering to think about how our actions are actively driving evolution in other species.
That's why understanding genetic variation is so crucial for conservation efforts. Without enough diversity, populations become vulnerable to any environmental changes.
Speaking of conservation, how do random events factor into all this? Like when natural disasters hit populations?
Well, those events can have huge impacts, especially in smaller populations. A single disaster might randomly eliminate certain genetic variants, dramatically shifting the genetic makeup of survivors.
And I imagine this is particularly dangerous for endangered species that already have small populations, right? They're probably more vulnerable to these random changes.
That's one of the biggest challenges we face in conservation biology. Small populations are much more susceptible to genetic drift, and they often lack the variation needed to adapt to new challenges.
You know what I find fascinating? How all these different mechanisms, mutations, natural selection, genetic drift, gene flow, work together like some complex evolutionary dance.
That's such an elegant way to describe it. Each mechanism plays its unique role in creating the incredible diversity of life we see around us.
Before we wrap up, let's talk about practical applications. How does understanding genetic variation help us in fields like medicine, agriculture, and conservation?
The applications are truly vast, from developing better disease treatments to breeding more resilient crops to protecting endangered species by maintaining their adaptive potential.
It really makes you appreciate how understanding these fundamental principles of life can have such far-reaching implications for our future. Evolution might seem like ancient history, but here's something wild. Bacteria can evolve antibiotic resistance in just a few generations, showing us that evolution isn't just about dinosaurs and fossils, it's happening right now in real time.
That's fascinating how quickly organisms can adapt. Speaking of adaptation, the early theories about evolution were pretty interesting, especially Lamarck's ideas about inherited traits.
Well, Lamarck's theory is like trying to explain how a smartphone works by saying tiny elves live inside it. He thought that if you worked out at the gym, your kids would automatically inherit those muscles. Let me tell you why that's not quite right.
Hmm. But you know what's interesting is that while his mechanism was wrong, he did grasp some fundamental concepts about how species change over time.
Exactly, he understood that organisms adapt to their environment, but he missed the crucial part about genetic inheritance. Like, if you dye your hair purple or get a tattoo, those changes won't show up in your children's DNA.
So, what really revolutionized our understanding was Darwin's work, especially his observations during the voyage of the Beagle, right?
Oh man, let me tell you about Darwin's key insights. First, he noticed something that seems obvious now, but was mind blowing then. All species produce way more offspring than can possibly survive. A single sea turtle might lay 100 eggs, but only one or two actually make it to adulthood.
Well, that really puts natural selection into perspective. It's like nature running millions of experiments simultaneously.
And here's where it gets really interesting. Darwin saw that within any population, you've got all these slight variations. Some finches had slightly longer beaks, others shorter, and these small differences could mean life or death depending on what kind of food was available.
The way those Galapagos finches adapted to different islands is such a perfect example of evolution in action.
You know what's wild? Those same principles are at work right now. Like, when we use antibiotics, we're actually creating intense selective pressure. The bacteria that happen to have resistance survive and reproduce, while the others die off.
That makes me think about how we often misunderstand survival of the fittest. It's not really about being the strongest, is it?
Not at all. It's more like, um, being the best fit for your specific situation. Like, during a famine, being smaller and needing less food might be more advantageous than being big and strong. Context is everything in evolution.
The way you describe it, natural selection sounds more like an editor than a creator.
That's exactly right. Natural selection doesn't create new traits out of nowhere. It's like having an incredibly picky editor who goes through millions of drafts, keeping what works and cutting what doesn't. The mutations in genetic recombination provide the raw material, but selection determines what stays.
Well, that explains why beneficial traits in one environment might not be so great in another.
Absolutely fascinating point. Let me give you a concrete example. Take sickle cell trait. In areas with high malaria rates, it actually provides protection against the disease. But in areas without malaria, it can cause serious health problems. It's all about the context.
That really shows how evolution isn't about achieving perfection, just finding what works in a specific time and place.
And here's something that blows my mind. Every living thing on Earth, from bacteria to blue whales, shares a common ancestor if you go back far enough. We're talking about a continuous chain of life stretching back billions of years.
That's such an incredible perspective on our connection to all life on Earth.
You know what's even more amazing? We can actually see evolution happening in real time with things like antibiotic resistance and pesticide immunity. It's not just ancient history. It's an ongoing process that shapes our world every day.
That really brings it full circle from where we started, doesn't it?
It sure does. And understanding evolution isn't just about looking back. It's crucial for facing modern challenges like antibiotic resistance, emerging diseases, and even climate change adaptation. We're all part of this incredible continuing story of life on Earth,